Hey guys, so we're going to start Unit C, the bio unit, and I'm going to regularly post the screencast videos. So the first thing that we're going to look at are microscopes. There are several structures to the microscope that you will need to know by the end of this week, and you will do a little matching quiz like you had done previously. You will drag and drop the terms and kind of label the microscope that I have for you. So these are all the structures of the microscope. But before we get into that, we're going to talk about the history of the microscope. And so there are some videos. You can click this live link here for history as to some of our discoveries and how we invented the microscope. So the scientists that you're going to need to know are Hans and Zachary Janssen. So this is a father-son team, and they came up with the first compound microscope. It looks like a telescope, and that's really kind of where it stemmed from. So in the 1500s, they came up with this compound microscope, and it's a compound microscope because it has two lenses, one on each side. So just like the lenses that we have in our glasses, if you wear glasses, lenses bend and refract light. So these two lenses, there was an eyepiece, which we would initially look through, and then an objective lens that would ideally magnify the specimen or the object that we were looking at. So this is what the first compound microscope looked like. Know that it was Hans and Zachariah Janssen. So this is a father-son team. Then later on in the 1600s, we have Robert Hooke. So Robert Hooke created the first light microscope. So he enhanced this first compound microscope and incorporated light to it. So that's this image on your bottom right-hand corner of the slide. So by incorporating light, the idea was that you could then create more of a contrast to the image. And he actually used three lenses for his microscope. One important note is that Robert Hooke actually coined the term cell that we still use today when we look at the cells of our body and of organisms all around us. So the way he came to this discovery is kind of cool, and we'll look at it later when we get to cell theory. I'll get you guys to watch this TED Ed video. You can watch it now and then rewatch it later if you want. But Robert Hooke ended up looking at cork. So you guys know cork, like at the end of a wine bottle, some of the things around you. Oh, I have cork right here, actually. This is cork, okay? And so he was looking at cork. So he was looking at dead plant matter, and he noticed that there were these openings, these chambers. And he called these chambers, these openings, cells, because he related them to monk cells, kind of like rooms that people stayed in. So we still use that term today. He didn't know it, but he was actually looking at those openings were where cells used to be, living cells, but they had died. So that's kind of cool that we still continue to use the term from the 1600s that he coined. So no, Janssen, first compound microscope. Robert Hooke incorporated light into his microscope and coined the term cell. And then don't get him confused with Antoine van Leeuwenhoek. Van Leeuwenhoek also created a microscope. He actually only used a single lens, but at this time, the lens that he used, he refined it, made it very clear in his cuttings, and he was actually able to look at living cells for the first time. So he was looking at bacteria, scrapings from within their teeth at the time, sperm, and so he was actually able to see the movement of cells and see that they were alive for the very first time. And he tried to get a term going, which was called animalcules. So he looked at all these different specimens. He looked at blood as well for the first time, and he coined the cells that he was looking at animalcules. But that term didn't really stick. So then we get to microscopes. So there are going to be a couple different types of microscopes that we look at. The main one is going to be this compound light microscope. It's the microscope that I'm going to get you to label, and it is what we use most often in a science laboratory, or even in universities. We often use compound light microscopes. They are still fairly expensive, but they're very limited in what they can do. So the magnification for using a compound light microscope is between 400 to 1,000 times the original size of that specimen. Now that's not really that big, enlarging the image that much in comparison to looking at an electron microscope. So electron microscopes are incredibly expensive. I only ever got to use one once in my whole university experience. And they are actually able to magnify an image up to 600,000 times the original size. So you're able to look at specimens, things all around us that are much, much smaller. And the images that they produce are really cool. So there are different kinds that we'll look into later. They project beams, electrons down onto the specimen, and they create or produce an electronic image onto your computer. And depending on what kind of electron microscope you're looking at, as a TEM or an SEM, you can actually also look at living species and produce 3D images of the stuff that you're looking at. So it's really cool. These microscopes are very large. They're about the size of you and I if we were standing upright. So they take up a fair part of the room. And they are very, very expensive. But the images that they produce, which we'll look at, are really cool. So for this week, we're looking at compound light microscopes. And this is an Amoeba Sisters video that I would like you guys to look at. They talk about what a compound light microscope is, as well as an electron microscope. Then looking at how to use a microscope. So typically around this time, we would bring in microscopes and you guys would get to look at pre-made slides and specimens. And so how to handle a microscope is really important because they are very expensive and they are fragile. So they have lenses, pieces of glass within them, and to replace them is very challenging and expensive. So we always have to handle them with care in how we hold them and move around with them, and what our workspace in the lab is going to look like if we're using a microscope is very important. So I do need you guys to watch this YouTube video, how to use a microscope. And they go over a lot of the general rules when in a lab and using a microscope, how to set up a microscope, and then how to focus in on an image on a slide as well. Moving down and moving on, we're going to look at the structures of a microscope. So by the end of this week, after you do some readings, after you watch the videos, I am going to get you to label a microscope. The image won't look like this, but very similar. So with a microscope, the first part that we're going to look at, and I'll highlight it, is the eyepiece. So all compound microscopes, there are variations to them, but they generally will look the same. So the eyepiece up here is just that, the eyepiece, what your eye looks through initially. And the eyepiece, when we get into magnification, it will magnify an image all by itself. So there's a little lens, there's a piece of think of like your glasses, okay? So there's a piece of a lens right through the eyepiece. And just this eyepiece alone will magnify your image 10 times the original size. As we look through the eyepiece, okay, you are going to have what we call objective lenses. So this microscope here is showing you only one, but typically there will be three objective lenses. And these objective lenses vary in how much they're going to magnify the image. So you get to choose which one you want, and they rotate. And so the objective lenses are another lens that help magnify the image. And so typically there is a 4 times objective lens, a 10 times objective lens, and then 40 times objective lens. And as you increase in the numbers, you increase in how much you're magnifying the image. So you have an eyepiece and then an objective lens. Then you'll have what we call the stage. So the stage is a platform that will move up and down, okay? And you are going to control whether it moves up or down. And the stage is where you're going to put your specimen or your slide. And so your slide is going to sit right on that stage. On the stage, typically you're going to have stage clips. And these stage clips just secure this slide, this piece of glass with your specimen on it, onto the stage. So it doesn't fall over, it doesn't move around on you. So you'll have a stage and then stage clips. Then at the very bottom of the microscope, we call this the base. So it's just the base, the bottom of the microscope. It's really good practice when we're carrying a microscope to carry it at the base, okay? So this is the base. It's just holding the whole entire structure in place. Then we'll have a light source below. So a light source is just that, it's a light bulb. Okay, it can be a plug-in microscope. So a lot of them you have to plug into an outlet in order for that light to turn on. There's also usually an on and off switch, or some are battery powered. And so you just recharge the batteries. The cool thing is at SPA, there are battery operated microscopes. And so you just have to make sure that they're charged and you don't have to worry about finding an outlet to use them. So you'll have your light source. Right below the stage, it's not labeled here, but there is a structure called the diaphragm. And diaphragm is like a little dial. You turn it one way or another. And what it does is it increases or decreases the amount of light that goes through to the specimen. So that will change the contrast or your ability to see the specimen. If you have a really thin slide, you might not necessarily want a lot of light going through there because it'll be really harsh on your eyes and you might not be able to see a lot of the structures. So you can dim the light. It's like a dimmer switch. Okay, so in some fancy living rooms or some fancy houses or rooms, you might see a dimmer switch where you can change the brightness of a light, and that's how the diaphragm works. So the diaphragm is going to be right below the stage. This is called the arm, okay? And so it also, you would say that it holds the objective lenses all in place and kind of gives it that structure. So this is the arm. Often when we're carrying a microscope, you'll always make sure you're holding it at the base and then you'll just support it with the arm. It's good practice just to support it at the arm. Do not ever grab a microscope just by the arm. The reason being is some older microscope actually have lots of gears along the arm here in order to help move the stage up and down. And I've done this myself. I've grabbed a really old microscope from a shelf and the top half is actually completely separated from the bottom. Because there are gears that help move it up and down and kind of change where the stage is going to be. So it's always good practice just to make sure you're supporting the microscope at the base and then, or holding it at the base and supporting it with the arm. Then there are these knobs. And so there are two knobs for every compound microscope. The first one is called a coarse adjustment knob, and it's going to be the big one. The coarse adjustment knob, this is the knob that changes whether the stage goes up or down. The coarse adjustment knob does large, drastic changes. So if you're really wanting to try and move around a specimen up and down, get it bigger or smaller, you're going to first use the coarse adjustment knob. Once you've kind of gotten your specimen into place, you are then going to use this smaller knob called the fine adjustment knob. And think of it just like when you're editing an image, if you're trying to fine tune it or sharpen the picture, you're going to use the fine adjustment knob in the same way. You'll have your specimen already in place. You should be able to see it, but to sharpen the image, to increase the contrast and to make it just a little sharper and get it more into focus, you'll use the fine adjustment knob. And what the fine adjustment knob does is it makes small changes in the movement of the specimen so you can get it into focus. One other structure that's not labeled on here is called the tube. And so from where you have your eyepiece to your objective lens, the structure that kind of holds both in place, we'll call that the tube. Okay. So this first structure, I'll just go from top to bottom. This is the eyepiece. This would be called the tube. Then we have, these two would be the same. We have the objective lens or lenses that vary in their magnification. We have our stage here. This is the diaphragm that will change the brightness of how much light is coming in from the light source down here. We have our base at the very bottom, and then if we come all the way back up, this would be your fine adjustment knob, smaller one, then your coarse adjustment knob, and then this is your arm. So I would like you guys to make sure you review the terms here. You do need to know the names of all the structures. You should be able to label the microscope, and then you should be able to define what the structures are all doing. Okay? The last part that I'll get into for this week is called magnification. And when we are looking at a specimen, it's important that we know how much it's being magnified. And when we're looking at a compound light microscope, it's fairly simple. I explained how the eyepiece, no draw for you. The eyepiece up here, if you were looking through it, the eyepiece magnifies an image 10 times, always. That number never changes. But then when we're looking at our adjustment knobs, you choose which one you're looking at. And the first one is 4 times. It's raining outside, hopefully you can still hear me. Is 4 times. So it's the least strongest lens. Then we have 10 times. And then we have 40 times the magnification. So depending on which one you use. Now, how we know what the overall magnification is we just multiply those numbers together. So what you're going to do is you're going to take the magnification of the eyepiece 10 times. Okay, 10 times. And you're going to multiply it by whatever objective lens you choose. So let's say you use the first one. You always start from lowest magnification and then get higher. So if we use the 4 times objective lens, okay, we're going to take 10, multiply that by 4, and so then your overall magnification of what you see and what you're looking at is going to be 40 times the magnification. Okay? If you're using the strongest objective lens, you would still take your 10 times for your eyepiece. You're going to multiply that by the 40 times objective lens that you're using. And so your overall magnification would then be 400 times. And so that's why I said compound light microscopes often will range from 400 times magnification up to 1,000. But most common microscopes that you and I use will max at 400. Not to say that there aren't some objective lenses that are stronger. Okay? So next week we'll look at electron microscopes and cells. This week, there are a few videos that I would like you to watch, and then there is a labeling quiz for your microscope that I would like you to complete for Mercs. Okay, hope you guys have a great week. Enjoy. Bye.